Hello everyone. In this video, we will discuss ion exchange chromatography in detail. Here is the contents which is to be discussed in this video. First of all, we discuss introduction of ion exchange chromatography, then principle of ion exchange chromatography and ion exchange resins, also classification of resins, and last we discuss application of ion exchange chromatography. Ion exchange chromatography is basically is a process by which a mixture of similar charge ion can be separated by using an ion exchange resin which exchange ion according to their relative affinities. Uh, Introduction. If we look at to the introduction of ion exchange chromatography, ion exchange chromatography is a type of adsorption chromatography. There is a reversible exchange of similar charge ion. Mostly similar charge ion like cation and anion can be conventionally separated by this technique. Many drug and pharmaceutical agents are weakly or strongly acidic or basic in nature. Hence, a mixture of similar charged substance can also be separated into pure components. Here is the diagram in which we can see anion exchanger stationary phase particle is attracted to positive surface and cation exchange stationary phase particle is attracted to a negative surface. Uh, Principle of ion exchange chromatography. This is by reverse exchange of ions between ion present in the solution and those present in the exchange resin. Uh, Cation exchange, the separation of cation using cation exchange resin. The cation to be separated are present in solution in exchange of similar ion present in cation exchange resin. A sided matrix, the exchange can be represented by the following equation. Here is X, which is the concentration of positive ion, plus RK, which is actually the concentration of negative ion. If we look at to the reaction proceed, here is some miswriting. It form R negative ion and X uh, X R X X it form X and RK negative ion solutions. The cation retained by the sided matrix of ion exchange resin can be evaluated by using buffer of different strength and hence separation of cation can be affected. Anion exchange separation of anion using anion exchange resin can be carried out. The anion to be separated are present in the solution in exchange for similar ion present in anion exchange resin. A solid matrix, the exchange can be represented by the following equation, which is the same equation, but here X represents the concentration of negative ion plus R plus Cl, which is actually chlorine. These are the exchange solution that form XR plus chlorine negative anions. The anion retained by the sided matrix of ion exchange resin can be eluted by using buffer to different strength and hence separation of anion can be effected. Here is the uh, diagram of ion exchange chromatography anion exchange. Here an example mix of amino acid which some are negatively charged and some are positively charged as shown. The flow of solution is in this direction. Immobilized cation surface is shown here. Binding of the negatively charged amino acid to immobilized cation surface, which is shown here. Separation of negative charge amino acid. It is also shown here while the positive charge cation is repelled by both sides and hence it is remain in the center and sit down while the negative are attracted to the positive walls of the chromatographic tube. Classification of ion exchange resin. So there are six types of these resin. The first one is surface of the resin. Source of the resin. The second one is chemical nature. The third one is functional group. Fourth one is structure type and physical property, and sixth chemical structure. Ion exchange resin should be have the following requirements. It must be chemically stable. It should be insoluble in common solvent. It should have a sufficient degree of cross-linking. The swollen resin must be denser than water, and it must contain sufficient number of ion exchange group. Here, it is divided into two classes. The first one is natural, cation, zeolites, clay, etc. are the example, and aniline is the dolomite is the example of aniline. Synthetic are maybe inorganic and inorganic or organic resins. Uh, Organic resin are a polymeric resin matrix. These resins composed of polystyrene set of exchangeable functional group and divinyl benzene cross-linking agent upper stability. The second one, according to the chemical nature, there are four types: strong cation exchange resin, weak cation exchange resin, strong anion exchange resin, and weak anion exchange resin. Strongly acidic cation exchanger, if you look at the example, it is sulfonic acid group attached to styrene and divinyl benzene couplomer. Weakly acidic cation exchanger are carboxylic acid group attached to acrylic and vinyl benzene copolymer. Strongly basic anion exchanger are quaternary ammonium groups attached to styrene and divinyl benzene copolymer plus nitrogen cations. If we look at to the functional group present in different ion exchange resins, a strong cation exchange resin contain sulfonic functional group. Weak cation exchange resin contain uh, uh, acetic aceto functional group or hydroxyl or thiol functional group. It may be phosphate functional group. Strong anion exchange resin is tertiary amine plus it may be tertiary amine and it may be amines. Weak anion exchange resin contain NHR and NH2. Structural type ion exchange resin, it contains four types. They are a follicular type with ion exchange film, porous resin coated with exchanger beads, macro-reticular resin bead, and surface sulfonated and bonded electrostatically with anion exchanger. Follicular type with anion exchange resins at 30 to 40 micro with 1 to 2 micro film thickness, a very low exchange capacity, and their anion exchange efficiency is 0.01 to 0.01 meter in ion exchange resin. Porous resin coated with exchanger beads. Its size is 5 to 10 micrometer. They are totally porous and highly efficient. Their exchange capacity are from 0.5 to 2 milli per gram exchange resin. Their molecular resin are bed and difference. Uh, Application of ion exchange chromatography is conversion from one salt to other. For example, we can prepare tetra-propyl ammonium hydroxide from tetra-propyl solution of same other anions. How state laundry detergents and water filters to produce soft water. Ion exchange is used to prepare deionized water, separate and purify metal, dealkalization, analysis and purification of immunoglobulins, separation of inorganic ions. Thank you for watching. In next video, we will discuss other type of chromatography.